2021-2022 CFAR external seminar series. And as you know, we hold these the first Wednesday of every month generally. Now going forward will be actually from 9:30 to 10:30, followed every other month by a science spotlight. So next month will be a science spotlight from the VA and the research going on there with HIV. The typical format for these seminars is we have an early stage investigator who sets the stage for the external seminar speaker. And what I will first do in today's ESI of the day is Dr. Matt Spinelli. Usually the ESI does research in the external seminar speakers area of interest. So Dr. Matt Spinelli does do research on PrEP, although today his talk is actually going to be focused on another topic. So, and then he will introduce our external speaker right after that. So today I'm really honored to actually start with the ESI of our day, which is Dr. Spinelli. Dr. Spinelli is an assistant professor of medicine in the Division of HIV, infectious disease and global medicine. Dr. Spinelli was an infectious disease fellow here and has been extremely productive, essentially went from infectious disease fellow to a K23 funded assistant professorship. And then very early on, after he got his K23, we were in the COVID pandemic, and Dr. Spinelli really responded to that call. He was very interested in the interplay of HIV and COVID in all dimensions, including does HIV lead to more severe outcomes with COVID, for example? Does HIV increase the susceptibility to COVID? He's also interested in immunologic responses of people living with HIV to COVID infection and COVID vaccinations. And finally, is interested in the social and behavioral aspects of the COVID response on people living with HIV. So put that all together, and Dr. Spinelli has an R01 and works very closely with other members of our division on all three of those aims. The clinic is really involved in aim 1. The link cohort, which is led by Dr. Peluso and Dr. Dakes, is really integral, and we work closely together for aim 2. And the social behavioral aspects, a lot of people in the division and at CAPS, including Malawi Johnson, are involved in the social behavioral aspects of this COVID time for people living with HIV. So actually what Matt is going to do today is talk about that interplay between HIV and COVID, and then introduce our external speaker. So thank you so much, Matt. Please take it away.
Thanks so much for that kind introduction, and for letting me talk about something other than PrEP. Like many people, my study site was shut down for most of the year and is just picking up. So my HIV and COVID data is perhaps a bit more interesting and timely at the moment. Great, let me go ahead and share my slides. Great, is that off the presenter view, the main view now? Good, great. So my talk is SARS-CoV-2 seroprevalence, clinical outcomes, and vaccination among people living with HIV. As we talked about in HIV grand rounds, most cohorts show similar or lower SARS-CoV-2 incidence for people living with HIV. However, initial data show lower COVID-19 disease severity based on hospitalized cohorts. That picture is updated somewhat, and now that subsequent large population-based cohorts have shown greater COVID-19 disease severity among people living with HIV, including in New York State and New York City, the UK, the Western Cape province, in South Africa, and also the more recent WHO data has shown that. In terms of vaccination, HIV subgroups have not, I would say, been well reported from the vaccine trials, although that's starting to change. We do know that responses to Hep B, yellow fever, and flu can be decreased among people living with HIV. So I'm going to be talking about the first two aims of the 1986 study, which is examining the interplay of HIV and COVID-19 at Ward 86 in San Francisco General. I'm not going to have time to talk about the socio-behavioral outcomes among people living with HIV, but save that for the future. We're going to be comparing people living with HIV at Ward 86 to adults without HIV, looking at COVID-19 disease severity, SARS-CoV-2 seroprevalence, and the immune response to vaccination. So I'm going to turn now to severe disease. Overall, in our hospital, we had 1,915 cases of SARS-CoV-2 infection when we looked just at Ward 86 and also the outpatient adult medicine clinics. And what we ended up doing was using a case cohort design to examine 1/3 of these cases, and we did in-depth chart review. In our analyses, we took into account this sampling fraction. We defined severe disease as a sustained oxygen requirement. And what we found when adjusting for age, sex, and race ethnicity, the risk of hospitalization was actually 90% higher among people living with HIV, and the risk of severe disease was 84% higher. So this is looking at people living with HIV at San Francisco General compared to outpatient adults getting primary care here. Some limitations is that we were not able to adjust for comorbidities, but we did find that there was 150% higher risk of severe COVID-19 in those with CD4 counts less than 200, suggesting that HIV is likely playing at least part of the role. So how about incidence of HIV and exposure? How about incidence of SARS-CoV-2 and exposure to SARS-CoV-2? What we ended up doing to look at this question was systematic testing of remnant lab specimens, which has been used in past epidemics, and also this epidemic when population-based serosurveys were not feasible. We ended up looking at remnant lab specimens from all people living with HIV in August through October. And we matched them on date of collection and age ±5 years to adults without HIV who were receiving laboratory monitoring in their primary care clinics here on campus. We looked at the anti-receptor binding domain, IgG, from BT Health. We adjusted for the test sensitivity and specificity. And among those with prior infection, we also tested for surrogate virus neutralization to kind of get at the immune response among those with positive tests. Overall, there were 955 people living with HIV included. What we found was that the seroprevalence was actually lower than I expected, just 4% among people living with HIV. And then those without HIV, it was a bit higher, almost 7.5%. That comes out to 52% lower odds of infection among people living with HIV, which I think is good news. But we wanted to get a sense of were there differences in the humoral immune response in these populations. So we went ahead and we actually looked at the quantitative IgG titers and also the surrogate virus neutralization test. What we found there was that there actually were 45% lower IgG concentrations among people living with HIV. We found something similar with the surrogate virus neutralization test. So I should add that these were adjusted for time since PCR confirmed infection, suggesting that there could be a decreased immune response among people living with HIV. So about this time, the vaccines were just being approved, and that got us concerned, would there be a reduced response to vaccination? So we were able to use the same methodology to save residual lab samples, this time from the month of April, post vaccination in most of our patients. And we then went ahead and matched them one-to-one to people without HIV. We only did mRNA vaccines, and so we matched them on whether they received the Pfizer or Moderna vaccine. Days since vaccination, they were the same birth sex and age ±5 years. We measured the same tests, and we defined non-response as the assay limits of detection, which were both 10 for both of these assays. And that this is a bit hot off the press, so I'm excited to share this with you now, but also concerned by what we found. So there actually were 43% lower IgG concentrations after mRNA vaccination. This is here looking in the upper left panel, suggesting that there were fewer quantitative antibodies that had been produced in response to vaccination. When we looked at actual non-response to vaccination, that's in the lower left panel here, there was a trend towards decreased response to vaccination, but it was not statistically significant. And interestingly, all of the non-response occurred in Pfizer BNT 162b2 recipients. Moving now to the right panels, and I should say that the red dots are the Moderna vaccine and the green dots are the Pfizer vaccine. In people who did respond with surrogate virus neutralization, there actually was no difference. But when we look at non-response here in the bottom right corner, there was decreased surrogate virus neutralization response to vaccination among people living with HIV. And you can see there was actually most people responded to the Moderna vaccine. So that was mostly driven by Pfizer vaccine non-response. Okay, see if we can move ahead. Okay, there we go. Then we went and looked at subgroups to understand who are the folks who weren't responding. For each 100 cell increase in CD4 plus T cell count, there were 28% higher IgG concentrations. And all 7 individuals with low CD4 counts below 200 did not produce an IgG response. Unsuppressed HIV RNA was associated with 86% lower anti-RBD IgG concentrations. And as we sort of had already discussed, the receipt of the Pfizer vaccine was associated with lower responses, 66% lower concentrations among people living with HIV. So what we found is that although there was fewer infections overall among people living with HIV, they did have higher rates of severe disease. And we think that's related in part to comorbidities, although our data suggests that low CD4 count is increasing risk. I think the lower seroprevalence could be related to either greater pharmaceutical intervention adherence or possibly a lower immune response. And the decreased vaccination response is concerning. I think it supports supplemental vaccination among some or potentially all people living with HIV. And I think if it's feasible, I would have a preference for Moderna given its greater immunogenicity, although I'd like to see that confirmed in more studies. And I think I'm out of time, but I'll just end with one quote. It's kind of like the HIV thing where you realize that things are different now with my body in the world. So I think it's actually helped me not be so afraid of COVID-19. And there's like knowing that I've survived HIV and now I can survive COVID. So that's a quote from one of our patients. These are my acknowledgements and thanks so much.
Thank you so much, Matt. I'd like to actually ask you to please stop the slide so I can see gallery view and invite anyone just for a couple. We just have room for a couple of questions before we move on to our main speaker. I wanted to just say, I think it's very intriguing what you showed, the difference between Pfizer and Moderna, as like you said, there's data sets in the Mayo Clinic system that are more rates of reinfection with Pfizer than with Moderna, and there's this idea of this shorter time interval with Pfizer. So maybe that will help us direct booster shots accordingly. Any other questions from the audience? What was the minimum time from, Peter Hunt asked, what's the minimum time from vaccine to antibody titer again?
Yes, sorry, I didn't release that, 2 weeks.
Yeah, so it may take 4 weeks until you reach maximal antibody titers. So it's possible that you could be, you may want to try a sensitivity analysis restricted to those over, with at least a 4-week lag.
I did do that, and it didn't make a difference.
Didn't make a difference.
They were matched on days since vaccination, so you would think the differences would be balanced. And then the other analysis also adjusted for days since vaccination.
Okay.
Moderna also has that higher dose. Any other burning questions? Otherwise, we will actually turn it over. That was great. And we hope to hear from you in the future about the other aims of this grant with Mallory Johnson. If I don't see any other questions, I'm actually going to ask Matt to please introduce our main speaker. Thank you very much.
Great, thanks so much. And just to follow up to Peter's question, I should say the median time since vaccination was almost 2 months. So most people were further out. Okay, great. So now switching to PrEP and to introduction of our esteemed guest. It's my pleasure to introduce our speaker today, Dr. Mina Hosseinipour, who's coming to us from the University of North Carolina in Chapel Hill. She's the protocol co-chair for HPTN-084, which is a study we're all very excited about, looking at cabotegravir versus TDF FTC PrEP among sub-Saharan African women, the scientific director of the UNC project Lilongwe in Malawi, and a professor of medicine at UNC. She earned her MD from Northwestern and completed her residency at Baylor and her ID fellowship at MPH at UNC Chapel Hill. She's lived in Malawi since 2001, which I think is pretty cool. And she's the scientific director of the UNC project there, where she's overseen many important clinical trials, as well as supporting capacity building programs for implementation science training. In addition to 084, she was also a protocol co-chair for HPTN 052, which was named by Science Magazine the Breakthrough of the Year in 2011. And I think so important, the findings of that study to the Undetectable Equals Untransmittable campaign, which has had a profound impact on my training and also on the happiness of my patients, I think I would say. So I will just add that we're very excited to hear more about her work on HPTN 084, which is likely to have profound impacts worldwide, particularly because prior prevention studies have shown adherence challenges. And I know I'm very excited to have this highly effective tool as part of our armamentarium. Her talk is entitled Injectable PrEP, the Game Changer for HIV Prevention. Question. So please join me in welcoming Dr. Hosseinipour to our CFAR seminar.
Thank you very much. Let me just get to sharing and hope that I do this okay. Are you seeing the right screen?
Looks great.
Okay, all right. Okay, then thank you very much. I'm actually really thrilled to be called esteemed. That's very nice. And thank you for the kind introduction and invitation. I need to forward. I might need to take off my video if it's, maybe I'm exceeding my bandwidth.
Make sure you're clicking on the, try clicking again on the Zoom screen if it doesn't, that helps. No?
All right, let me, oh, I did something bad. Okay, let me try again. Sorry.
If anything goes wrong, you can send us your slides and we can advance them. But
well, we practiced the first 15 minutes. I was doing perfectly, and then what happens? How do I not have this down yet in this Zoom era?
what we're excited about is this is unpublished data, right? So
well, I won't say that.
Okay. Presented, but not yet.
Presented, not yet published. I think that HPTN would have my hide if I presented informally to UCSF CFAR, but first. Okay, do you all see the right screen?
Yes.
Okay, all right, I think I have it now. All right, sorry for that. I've taken some time working through, unfortunately, technical issues. So I'm going to acknowledge from the beginning what the data I'm presenting are from the 2 published papers for 084, and then the HIV-R4P presentation from January 2021, and then the laboratory analysis that was presented at IAS in July. So some things you might not have caught since IAS, we're busy with a lot of things. So we all know that antiretroviral pre-exposure prophylaxis can reduce HIV acquisition, and the primary barrier to success has been largely adherence to the daily oral dosing. And so novel long-acting products like cabotegravir are intended to simplify PrEP use by improving adherence and allowing people to then meet their prevention goals. So we know there are many advantages potentially to an injectable product. First, it's stated to be a preferred product by many populations at risk. The idea of infrequent dosing is very appealing. This addresses adherence issues, and it can make it very discreet because you don't have to have a bottle of pills. Disadvantages are that once you give it, it can't be removed. This has the potential for prolonged side effects or potentially resistance in a tail phase due to its long pharmacologic tail. And new data is suggesting that diagnosing HIV using the typical HIV testing algorithms can be made more complicated by long-acting cabotegravir. I'll present some of that today. So this is all based on the HPTN 083 and 084 studies. Both of these studies had a primary objective to evaluate the relative efficacy of cabotegravir injectable versus daily oral Truvada for prevention, and as well looking at the relative safety of the products. Here's some details about each of the studies. 083 was a study in cisgender men and transgender women. It had a near global distribution as to where it enrolled. It was enrolled in 4 continents. It began in December 2016, and it was a non-inferior design because there was a point estimate of how effective Truvada should be with respect to prevention efficacy. For 084, it used the same products. It was initially projected to enroll 3,200 women. It enrolled exclusively in Southern Africa. It began in November 2017, and it was designed as a superiority trial. Both studies were targeting the primary populations at risk for their setting and really focused to have the most at-risk individuals based on epidemiology of the settings. Certainly individuals who would meet criteria and benefit from pre-exposure prophylaxis. So 083 eventually enrolled 4,570 men. They met their targets of having greater than 50% under age 30, greater than 10% transgender women, and greater than 50% of the US enrollment to be persons of color. And the 084 study enrolled 3,224 women. They were assessed for their risk using a modified risk score, which is a score that addresses age, partner characteristics, alcohol use. And it was increased to a risk score of 5 to ensure that the expected incidence would be met in the study. The design of both studies is similar. There is a step 1, which was an oral lead-in phase to evaluate tolerance prior to injection. A step 2, which was the active product injection versus placebo injection, and corresponding to which arm you were on. And then step 3, which was a 48-week open label Truvada state, where this was to cover the tail. So specifically, if you were on the cabotegravir arm, you took cabotegravir pills, Truvada placebo, you got cabotegravir injection, and Truvada placebo in step 2. And if you were in group B, you got the cabotegravir placebo with active Truvada, and you got injection placebo and Truvada active. And the design for 083 is the same. You can just decide which schematic you think is more beautiful. In terms of efficacy, how well did these work? Notably, both studies had the same data safety monitoring board and were evaluated every 6 months. And both studies were stopped early for demonstrated efficacy. So these are the 083 results. And in the 083 study, there were 13 infections in the cabotegravir arm and 39 in the Truvada arm, related to an incidence of 0.41 for cabotegravir and 1.22 for Truvada. The hazard ratio was 0.34, which favored cabotegravir and resulted in a 66% reduction in HIV acquisition compared to Truvada. But this is also a low incidence in the Truvada arm, so we can recognize that both have worked well. For 084, we saw a pooled HIV incidence of 1.03. In the cabotegravir arm, there were four infections. This related to a HIV incidence of 0.2, and in Truvada, the incidence was 1.86. So when I first saw this Kaplan-Meier curve, I just went, wow. It's not quite as good as the COVID vaccine, perhaps, but it relates to an 89% lower risk of HIV infection in the cabotegravir arm compared to Truvada. And since I focus mostly on research in women, I made this quick graph that looks at studies that have reported for HIV prevention in women in Africa in the last few years. And here's on the far left is the cabotegravir at its 0.2 incidence. And the Truvada arm also lower than what we've seen in the ECHO study, Imbokodo vaccine study, the Hombo vaccine study, and the AMP protocol. So you can see that when we say that 084 has the potential to be a game changer, it's because really no infections, like so few infections were taking place compared to what we might have expected. So that's why it's clearly one of the most exciting things that has come out in HIV prevention, which I would say since 052, but I may be biased. With respect to adherence, this was an important component that the studies evaluated because it has been so linked to efficacy of the products. So both studies had subsets in which they tested the values of tenofovir levels to understand adherence. So this is the 083 data, and you can basically, to make this simple, anything in green suggests it was pretty good adherence and should prevent infection, and anything in yellow, orange, or red is maybe less than ideal and might not be fully protective. But what you see is that 76% of participants overall in this subset had detectable levels at a positive level, suggesting good adherence. It did go down somewhat over time, but remained relatively high if you compare it to the 084 study. Here, overall, 64% had detectable tenofovir levels, and only 46% were detectable in what would be suggestive of daily use. And over time, it started off fairly reasonable, but really dropped off by week 57, which was the last time point that we've evaluated as of now. So adherence was definitely an issue. And this is the 084 data in terms of injection coverage, which shows that essentially at each of these intervals, it was close to 90% coverage of participants getting their doses. So that's certainly a much higher coverage level than was achieved with the daily oral tablets, and likely explains these really remarkable efficacy results. So with respect to safety, you've got a really highly effective product. You want to know that it's safe. And so with respect to grade 2 adverse events in 083, there were some slight differences in terms of the different arms. So the Truvada arm had slightly more creatinine clearance that decreased. Not entirely clear about the nasopharyngitis, why that should have been different, but it was more common in the cabotegravir arm. Some increased glucose was noted in the cabotegravir arm, and fever was also slightly more common in the cabotegravir arm. Looking at that same spectrum of grade 2 or higher adverse events, in 084, we didn't actually see any differences in the two arms that were notable for 084. In terms of where we did see differences is injection site reactions. So in the 083 study, there was over 80% of participants reported a injection site reaction of some level, and it decreased over time. And there was only a small number of patients or participants that elected to discontinue cabotegravir due to intolerance to the injection site or to the injections, and having injection site reaction that was uncomfortable for them. So in the 084 study, though, we saw far fewer injection site reactions. 25% of the participants had it at the first visit, and then it really drops off to very low levels over time. And no participants in the 084 study discontinued injections due to injection site reaction. Weight gain has been an issue with integrase inhibitors for treatment. So this was something that was evaluated in both studies. Here in 084, we see that there is an initial increase in weight of 0.42 kilograms in the initial period. And then there's a steady increase over time in both arms, such that by the 3 years since enrollment, there actually doesn't end up to be a statistically significant difference in weight. In 083, something similar is seen. There's an initial weight gain in cabotegravir, and there's a slight weight decrease in the initial 41 weeks of treatment with tenofovir FTC, so that you see a statistically significant difference in weight at 41 weeks, but then it tapers off so that the weight gain in the subsequent time periods is not statistically different. Previously in 077, there wasn't a difference that was seen over 41 weeks, but that was a much smaller study. So in conclusion, with respect to efficacy, we know that both agents evaluated in 083 and 084 were highly effective. 084 had the pooled incidence of 1, and 083 had a pooled incidence of 0.81. Cabotegravir was superior in both studies. So 89% lower risk of acquiring HIV in 084 and 66% lower risk in 083. And the 8-weekly injection likely provided a significant adherence advantage over daily oral Truvada. So for safety, both products were safe and well tolerated, with few differences in grade 2 or higher adverse events, apart from injection site reactions, which were more common in the cabotegravir arm. These were generally mild, associated with pain, and occurred at the first injection and then decreased over time. Men experienced more injection site reactions than women. And also with related to safety, the initial weight gain occurs slightly more in cabotegravir, but there are modest gains in weight in both arms over time. So we can conclude that CAB is safe and effective. So it's really panning out as quite the game changer. But do the breakthrough infections make us think otherwise? So we have now, for 084 and 083, we've now evaluated the pharmacology and the resistance profiles of the seroconversions. So in 084, we have 4 incident infections. And they follow 2 patterns. For B1, this participant was in the oral phase, and then they were tested positive and found before they went to the step 2 injection phase. We subsequently determined that the levels of the oral agent were low in this particular individual. The B2 is a person who took the oral phase, became pregnant, began open label Truvada, and then had a bit of a lapse, and then was later found to be infected. So they did neither of these individuals received an injection. The other group reflects individuals who did receive injections. D1 is an individual who received 8 injections, but in the dashed lines here, you can see that some of them were delayed. And in particular, there was a delay between her last injection and when HIV was detected. And then the other individual, D2, appeared initially to have quite good spacing of their injections, but when we went back to evaluate baseline samples, it turned out that this individual was infected at enrollment and was not detected that they were an acute case at enrollment, and did go on to proceed to get injections. So of these individuals who ever received injections, actually in all 4, there was no integrase resistance that was detected. In 084, we had 36 incident HIV infections. They were, actually, let me go back to this to just say that this little green emoji is when the sites detected the infection and when we felt it was to occur. So this being identified to be an incident case was something that was sort of unexpected, but it's different than what we'll see in 083. Here for the tenofovir cases, there were 36 infections, and 35 out of 36 did not have levels that would suggest adequate adherence to the product. The concentration showed poor or inadequate adherence, which likely explains why these were breakthrough cases. Infections were detected near real time in terms of their participation in the study. For 083, there were 13 incident infections, and I can't really deliver these details with the zeal of the 083 chair, Rafi Landovitz, but I think that the main points to take away from these infections are that 4 actually were incident cases at baseline. And among a large number of these cases, you can see that the emoji of when HIV was established was often not at the same time as, like, they were able to get injections afterwards because the site wasn't detecting the infection in real time as we'd hoped with the algorithm. And similar to 083, the adherence of these individuals was suboptimal in many cases, to explain why there were breakthrough infections in the Truvada arm. So extended testing identified infections that were earlier than were identified by the sites. So there was delayed diagnosis of HIV, which allowed continued cabotegravir injections. The virus loads were often very low at the first positive visit, and there was often prolonged period of viral suppression after infection, which complicated diagnosis. And antibody expression was diminished or delayed. And in some cases, we even saw that the RNA and antibody test would revert between negative and non-reactive early in infection. So what this lets us know is that while it looks like a game changer, we may need to change some coaching strategies to reach the full potential. So missed acute infections, we would have likely found all of these had a viral load been done to exclude acute infection prior to initiation of any PrEP. We did the viral load at screening, but it wasn't repeated at enrollment. And so we would have likely found those infections for both studies by doing a viral load. Late diagnosis for the follow-up studies of 083 and 084, we're going to include an HIV RNA as a part of the diagnostic algorithm to see how this improves accuracy and timeliness of diagnosis. There was emergence of resistance. I think I forgot to mention that, that 5 of the 9 cases, or 5 of the 13 cases in 083 showed integrase resistance. So their subsequent treatment with a integrase inhibitor may be compromised. So this is obviously not a result that we want. And then we haven't really been able to evaluate the tail. We haven't really had sufficient information to say what's going to happen with the tail because of the way the studies were rolled over into open label extensions. But as of now, we haven't seen emergence of resistance in that group of individuals. Other populations we need to think about, and we don't yet have results. The 08301 and 08401 are adolescent studies. We know that adolescents are at high risk. So several of the sites that participated in 083 and 084 are doing these companion studies. And the goal is that we can have concurrent licensure of cabotegravir for adolescents and adults in close proximity, because typically in other studies, adolescents have been left behind. Another area that we are continuing to evaluate is pregnancy and the role of cabotegravir use in pregnancy. So for 084, we identified 50 pregnancies in the study. They were essentially close to the same, slightly more in the cabotegravir arm, but we did manage to maintain a very low incidence rate because we had very strict contraceptive requirements. So we have outcomes for a small number of pregnancies. And I think the point I would like to make is that we need more information about pregnancy outcomes. And so one of the elements of the open label extension for 083 and 084, or for 084 only, because only 084 individuals can get pregnant, is to really characterize pregnancy in a better way. So let's go on to what are some of these next steps. So I'm going to highlight 084 that we've specified. Our objectives are to continue to evaluate incidence of HIV as we combine the blinded, unblinded, and open label period, so that we'll have a larger number of HIV seroconversions and have a better understanding of implications of seroconversion on cabotegravir. We want to evaluate open label cabotegravir with and without an oral lead-in. We believe that it's critically important to simplify the administration of cabotegravir for program implementation. And the initial data suggests that the oral lead-in is probably not necessary due to the very low rate of side effects or adverse events that were experienced with oral lead-in. So this is the key element of both 083 and 084, is to evaluate with and without an oral lead-in. We want to evaluate acceptability of open label cabotegravir, estimate incidence of pregnancy among participants, and also evaluate safety and infant outcomes among the pregnant participants, including more in-depth pharmacokinetics of cabotegravir in pregnant patients and in their infants with respect to how much breast milk exposure they have. So we have a lot that we want to do in open label extension of 083 and 084. Both of these are now, 083 is in the field with its open label extension, and 084 has gone to the ethics committee. It's just newly been released. So hopefully all of those will allow us to evaluate the new algorithm and also get more information on pregnancy acceptability and whether the oral lead-in can be successfully removed from this prevention package. So essentially participants will choose to either begin cabotegravir long-acting or Truvada. They'll be followed for 48 weeks. They can switch from Truvada to cabotegravir, and they can either take the 4-week lead-in or they can start injections immediately, and most visits will be every 8 weeks. The contraceptive requirements that were so very strict for the original version will be relaxed, and participants will not be required to use any form of contraception, and this will allow us to have more knowledge about what women want with respect to becoming pregnant while taking PrEP, and also more outcomes related to pregnancy. And we know this is really important to the women right now because so many of them want to start childbearing, but they're still in the study. So this, I think, is really important for the study. And I think broadly, when we think about how this is going to roll out or how we're going to move it to the next phase, we really need to do more research on sort of preferences about what they might want to receive, how they might want to receive it, and so forth. So these are examples of formative research that really led us to know that women wanted to be using injectables. It was very much an important, most commonly stated preference for women. Implants were also desirable, and I won't get in it today, but we know that implants are a next phase preventive strategy that we hope to move to the field very soon. And I think another thing, this is a study that really looked at a discrete choice experiment that looked again at what adolescents and young people wanted in South Africa. It really highlighted that to them, the frequency of how often you have to get a dose of whatever prevention product you have is what's most desirable to them. So right now, when we only have, say for women, oral PrEP, a dapivirine ring, and cabotegravir, you'll see that this every 2 months for the injection is very appealing to young people, with the highest number of people really focused on how long and how few times do I need to come to a clinic. And as we look at these preventive comparisons, many of these are now going to be available at many of the sites where we're delivering PrEP. And so counseling messages that really highlight what are the pros and cons of each method will become something that has to be embedded within the programs if all have been chosen to be adopted for a country program. So I think that this type of research will be critical. So in conclusion, I'll say that cabotegravir is safe and effective in men, transgender, and cisgender women. The HIV diagnosis algorithms will require refinement to identify infections as promptly as possible to prevent emergence of integrase resistance. The expansion to include pregnant women and adolescents is forthcoming, and we will get a real feel for what women want when they're allowed to go without family planning as a part of their prevention strategy. We need to focus on simplification of how Truvada or cabotegravir can be delivered by eliminating the oral lead-in if this is well accepted and found to be safe. And we need lots of implementation science that will evaluate the comparison of prevention products going forward. And so I'd like to acknowledge mostly the funders of the 084 and 083 team and the amazing teams that participated in both of these landmark trials. And I'll conclude by saying, yes, I think it's a game changer.
Thank you so much, Mina. That was really fantastic. And I'd like to open it up to questions for you. You know, I'll start with, you presented data that we saw before from both from the HB10-083 CROI 2021 data on the laboratory resistance data, and then you showed no integrase resistance in the 4 failures in HB10-084. And I wondered if you thought there were pharmacokinetic differences in women that have already been reported that would explain possibly why the MSM data showed both more failures and both more resistance.
Well, from 077, we definitely saw that women had a longer tail and also could actually space out their injections almost to remain above the therapeutic levels to 12 weeks. So definitely for women and those individuals with higher body mass index, we know that those are associated with sort of longer periods before you start declining your levels. So I think that that may be playing a role. There may be also a difference in penetration in the target tissues, vaginal versus rectal, that might also help explain some of the differences.
And I mean, it does, make one wonder about the every week's watching men a little more closely, MSM and transgender women. Okay, so should we, I would love to please see if anyone could either raise their hand, put it in the chat, unmute yourself, and go ahead and ask your question. This is really an exciting opportunity to have such, hot off the press data presented to us, and see if questions about this. Someone is trying to speak.
I think Susan has her hand up.
Hi. Oh, thank you. Sorry, Susan. I'm
That's
so
okay.
On gallery. Please go ahead.
I can't see everyone, but I do see that.
That was a beautiful presentation. I had 2 quick questions. One is, given the problem with diagnosing HIV infection in people who've gotten cabotegravir, did you go back and test all of the specimens for all of the participants who were in the cabotegravir arm to see if there were any undiagnosed infections? And the second question is a little bit separate, which is we've got now really fantastic data on protection against sexual transmission. And I'm wondering if you know if anyone is planning to study this for injection drug use. Thanks.
For the first question, no, we have not tested all of the cabotegravir participants to see if there are further undiagnosed cases. I don't think that's necessarily in our plan right now, but we, I guess, can look at whether there might be. I know the other thing that we haven't reported out is how many actually false positives then came back into resume treatment, to sort of have a real feel for when you get a positive test, how often is it this masked seroconversion due to cabotegravir, and how often is it a false positive? Because both definitely occurred, but we still need to do more to evaluate that, at least from the 084 perspective. Like I said, Rafi would know everything about 083, but I can't command his knowledge of that study. And to my knowledge, there isn't an IV drug population under study, but I could stand to be corrected. There are sometimes things that slip under my radar, but I don't know of one right now.
Thanks.
Steve, you had indicated you had a question, Steve Deeks.
I mean, so great presentation. I've always been intrigued by these weight gain stories, and I think studying this in people with HIV has always been complicated because people are switching on or off of bad drugs, and so studying it in people with HIV is more informative. And I'm wondering, so it looked like there was an increase in weight gain among those who got cabotegravir early, really quickly, suggesting it's doing something. I'm wondering if what the two questions is, have you guys seen any outliers? Like, is it, you looked at the median, but are there a subset of people who are getting massive weight gain on these drugs who don't have HIV? And do you have any idea what the mechanism may be for this stuff?
I'll admit that that was an analysis that I wanted us to look at to see if it's that higher BMI individuals like gaining more kgs, or if it's like the lower weight individuals that are having it. And it doesn't seem to have a trend that I can decipher from our initial evaluation, but I think that's an area that we'll look at. And I don't really have a great mechanism of action. In 083, those who were taking Truvada actually lost weight a little bit, which accounts for the difference, and maybe sort of GI side effects that maybe were more prominent, although not necessarily reported in our data. I know that there might be that subset that had some weight loss from that, but I'll admit, I don't have a good understanding of why it should be an early weight gain and then everybody catches up. I will say that our weight data in 084 may be complicated because we have had a number of individuals who had COVID-related social harms where they didn't have adequate food. So like we've had these weight loss issues in some participants as that sort of panned out. But I think it's an area we'll continue to evaluate more on and try to understand.
Is this, a real quick follow-up question, is this issue in my clinic, some people are pushing back a little bit on these drugs because they've heard about this weight gain with integrates. And I'm wondering if this is an issue for the community in terms of uptake of this approach with people with PrEP.
I imagine that, I guess as we begin our counseling messages, we will include that there may be a slight increase in weight when starting the product, and it'll sort of, you know, be similar to the Truvada over time. I mean, I guess all of us are sensitive to 1 kg, but in the big scheme of things, I don't think that the participants were becoming massively overweight. So I know we all like to be trim. So body conscious people don't want any extra kgs. And I, as a cyclist, don't want an extra kg, for example. So
I think last question, Mina, from Dr. Havlier.
Oh, hi, Mina. That was just a fantastic talk. Thank you. Great to see you. I just was wondering, I know integrase inhibitors have been used in kids, and I'm so glad you're doing what you are in the extension, because it's the only practical use of these drugs, right? Because women are going to get pregnant. Is there anything like, do people have any concerns at what's going to happen to the infants at all?
Well, we don't have a ton of data on infants yet because there have been relatively few pregnancies in the treatment and in the prevention studies. Like, we're really looking at handfuls of cases. So even with the open label extension, we don't think that we'll be able to, see any increased risk of congenital anomalies, for example, because but it will be very controlled, and we'll have a good understanding of how much cabotegravir is being transferred to the baby. But we don't expect that there should be any problems based on sort of the initial sort of animal phase data and what we have available to date.
Good. Oh, that was really my question. Okay, thanks.
Mm-hmm.
Thank you so much, Mina. That was a fantastic talk. Led on Diane's HIV grand rounds this morning. This is really the exciting things to talk about in HIV. So thank you so much for your participation. We really appreciate it. And IAB members, it's actually a different link. So please go ahead and go over to the IAB member, IAB meeting. Thank you for joining, and everyone, thank you for joining this inaugural seminar.
Thank you. Bye.
Bye. Thanks again.